Welcome to the cBioPortal tutorial series brought to you by the Genomic Education team at the Jackson Laboratory. This video will show you how to locate the names of the genes found to be mutated, also called gene variants, within a single study on the cBioPortal. Because the cBioPortal is a database for cancer genomics data, the studies include data on gene variants identified in the patients within the study. In other words, this is a list of which genes were found to be mutated in the cancer tissue of the patients included in the study. To locate these genes, find the table called Mutated Genes. Like in this pancreatic cancer study, we see the Mutated Genes table here on the right. There are 4 columns included in this table. The first column on the left is titled Gene. This is the gene name. Genes are given abbreviations of a few letters with or without numbers. You can see the first gene listed here is KRAS, or KRAS. The second column is number MUT, or mutation, standing for the number of mutations found in the samples in this study. The next column, titled Number, is the number of samples in which the mutation was identified. In this study, one sample came from one patient, so therefore you can think of this number as the number of patients. And finally, the column on the very right, labeled FREQ, or frequency, is the percentage of samples or patients with a mutation in this gene out of the total number of samples or patients within the study. Let's go through an example. The first gene listed in the table is KRAS, and we can see that there are 103 mutations identified in the samples that went through genetic profiling in this study. There were also 100 patients having KRAS mutations. So wait, these numbers don't match. Because each patient provided only one sample, we'd expect if there were 100 patients with KRAS mutations, then there would have been 100 mutations identified. Therefore, in this example, it means that some of the patients in the study had more than one mutation in KRAS. However, if we look at the frequency, we would find that 91.7% of the patients have KRAS mutations, as this number is calculated by dividing 100 by 109 patients in the study. Another feature of the Mutated Genes table is the search bar. Because there are typically hundreds or even thousands of genes included in this table within a study, in order to find a particular gene of interest, you can use the search bar at the bottom of the table. Say we were interested in the gene PCLO. By typing the gene name into the search bar at the bottom, the table is filtered, and now we see only the information for PCLO. Thank you for your attention. To learn more about the cBioPortal and its features, continue to watch videos in this tutorial series. For other resources from Jack's Genomic Education, find us on our website.